Chapter 17 takes us through the steps that the cell takes to manufacture a protein. Starting with the information in the DNA, it's going to go through two important processes: transcription and translation. Transcription is the synthesis of an RNA molecule that contains the information of the DNA. The RNA molecule is then taken to the cytoplasm, and in the cytoplasm it is going to be translated by the ribosomes. And the translation of the messenger RNA by the ribosomes will then give rise to the protein. An overview then of the principles of transcription and translation. Transcription refers to the making of an RNA molecule, and it is going to use DNA as the template. So DNA is going to give the example, and the RNA is going to be copied from DNA. So transcription goes from DNA to RNA. For genes that code for proteins, the product of transcription is going to be mRNA. mRNA stands for messenger RNA. The process of translation refers to the making of a polypeptide using information that comes from the messenger RNA. So it's the next step in the pathway. We go from messenger RNA to protein. This step is performed by the organelles that are called ribosomes that are found in the cytoplasm of the cell. So transcription occurs in the nucleus and translation happens in the cytoplasm of eukaryotic cells. Transcription and translation are two very important and basic processes that happen in both prokaryotic and eukaryotic organisms. In prokaryotes, in other words in bacteria, transcription and translation can happen basically at the same time, concurrently. So the process of translation can begin before transcription has even finished. And the RNA transcripts, the molecules of RNA that are formed in transcription, are not processed. They're immediately ready for transcription. There are no separate compartments in a prokaryotic cell, which makes it possible for a ribosome to attach to an RNA transcript while the transcript is actually still being formed. In eukaryotes, on the other hand, the processes of transcription and translation happen in different compartments. Transcription occurs in the nucleus and translation occurs in the cytoplasm. And also the RNA transcripts are going to be processed before they leave the nucleus. So in eukaryotes, there is necessarily a gap between transcription and translation. You get transcription, processing of the transcript, and then you're going to move the transcript into the cytoplasm, and then you have translation. This picture illustrates the fact that there is no compartmentalization in prokaryotes. We're looking at a bacterial cell, and we see the DNA molecule in the cell. The process of transcription is going to generate a messenger RNA molecule, and that messenger RNA molecule is going to be translated by a ribosome, which is then going to produce a polypeptide. All of it happening basically in the same space. This slide illustrates what was being described in the previous one. So we see on the top an electron micrograph, and in the bottom the interpretation. Let's start with the bottom because that is clearer, easier to explain. The blue line that we see here is the DNA molecule. So that is the template that is going to be transcribed. It is being transcribed by RNA polymerase, and there are 4 copies of RNA polymerase moving along the DNA. As each of them moves along the DNA, they produce an RNA transcript in red. So here is the RNA and the inset in red. But notice that even as the RNA is being made, there are ribosomes associating with the RNA, and the RNA is getting transcribed immediately. So each of these little brown balls is a ribosome, and the protein is coming out of the ribosome. So we have the process of translation occurring while the process of transcription is still taking place. On the top we see the actual picture of what is happening. The DNA is very faintly visible here on the top. If I trace that line, it's here. The RNA that's being made is trailing off of the DNA, and the ribosomes are the little dots that are associated with the individual RNA molecules. The proteins are not that clearly visible in this picture. Maybe you see a little piece of a protein here and another piece over here. Those are coming from the ribosomes. In this slide, we see how the situation is in a eukaryotic cell. And if you compare it with what we saw with the prokaryotic cell before, we notice that the eukaryotic cell is compartmentalized. In other words, there are separate compartments in the cell. We see the nucleus, and then outside of the nucleus is the cytoplasm. The process of transcription happens inside the nucleus. So we've got the DNA template in the nucleus, and transcription is going to give us a pre-messenger RNA, not even a mature messenger RNA. That pre-mRNA is going to go through the process of RNA processing, and RNA processing is going to give us a mature messenger RNA. That messenger RNA, that mRNA, is going to be taken out of the nucleus into the cytoplasm, and there it's going to be translated by a ribosome, and that ribosome is going to give rise to a polypeptide chain. So the process of translation is happening in the cytoplasm outside of the nucleus. The general flow of information in a cell then goes from DNA to RNA to protein. This concept is so universal that it has become known as the central dogma. There are a few exceptions to it, but you could look at it as a rule that is valid in almost every situation. In the step of translation, when we're going to be changing from messenger RNA to protein, we're going to use a set of information that is known as the genetic code. In other words, the information in the messenger RNA needs to be interpreted to make the protein, to make the polypeptide chain. Codons in the messenger RNA are going to be used to specify amino acids, but it takes 3 nucleotides to make 1 codon. In other words, it takes 3 nucleotides of RNA, of messenger RNA, to specify 1 amino acid. Notice that the code, the genetic code, is written in mRNA triplets. In other words, you're going to see U's in the code and not T's. only one of the two DNA strands is going to be used as template to generate the code. So we're looking at the illustration here at the two DNA strands, and one of those two strands is going to become the template. In this case, the template is the upper strand, and you're going to generate an RNA strand that is complementary to that upper strand, and that is going to be identical to the lower strand, except for the fact that wherever there is a T in the DNA, in the RNA you're going to have a U. But otherwise, these two strands have an identical sequence, and they also have the same orientation, with the 5' on the left and a 3' end on the right. So notice, so T becomes U, but then you've got G, G, T becomes U, and then again G, G, C, T becomes U, C, and A. If we look at the codons in this messenger RNA here, then it takes 3 nucleotides to make a codon. So you've got UGG forming the first codon, and that codon specifies the amino acid tryptophan. The next codon, UUU, specifies phenylalanine. GGC gives us glycine, and UCA gives us serine. In the last slide, we saw that the non-template strand of the DNA, the one that is not getting transcribed, was essentially identical to the messenger RNA transcript. The non-template strand is therefore called the coding strand. It has the same orientation also as the codons in the messenger RNA. The genetic code is written in messenger RNA triplets. In other words, the code is going to have U's and not T's. There are 64 different codons in the genetic code. What that means is that there are 64 different combinations of 3 nucleotides that will specify different amino acids. Of those 64 codons, 61 codons code for the 20 different amino acids. That implies that the genetic code is redundant. Another word for that is that the genetic code is degenerate. That means that more than one codon codes for the same amino acid. Notice that it does not mean that more than one amino acid is coded for by the same codon. If we take an example, say the codon CAU and CAC, oh, I can't write those letters, can I? CAU and CAC both code for the amino acid histidine. But CAU only codes for histidine and CAC only codes for histidine. They don't code for any different amino acids, but histidine is coded for by both CAU and CAC. The genetic code is also non-overlapping. That means that the codons are read without spaces or pauses and no overlap with each other. So you start in one specific position, which is going to establish your reading frame, and you just keep going from there without skipping and without overlapping. So we said there were 61 codons that code for amino acids, and then there are 3 codons that don't code for anything, and those are called stop codons or termination codons. This table shows us the complete genetic code with all 64 codons, all 64 possible triplet combinations. The triplets are shown here, the 3 letters, you see them in groups of 3, and then next to the triplets you see this 3-letter abbreviation that represents each individual amino acid. So this PHE stands for phenylalanine, leucine, isoleucine, valine, and so forth. Notice that there can be several codons that code for the same amino acid that we mentioned already. For example, you have here all of these 6 codons code for the amino acid leucine. If you look at one of those codons individually, though, each codon only codes for one amino acid. So CUA only codes for leucine and for nothing else than leucine. But leucine is coded for by all 6 of these codons in this group here. This green codon, AUG, is special because it is the start codon. All polypeptides start with the amino acid methionine when they are initially synthesized. Often the methionine is later removed, so mature polypeptides don't always start with methionine. Then there are these 3 codons here, UAA, UGA, and UAG, the stop codons. When the ribosome comes across one of these, then it releases the messenger RNA, and it also releases the polypeptide that it was working on up to that point. So these are stop signals. I made a little animation to show you how to interpret the genetic table. To access that, please click on the link in the other comment on this same slide. The genetic code helps us translate messenger RNA codons into amino acids. The 64 mRNA codons are arranged in a table. As you can see, each codon consists of 3 mRNA nucleotides, or a triplet. The first letter of the triplet is represented by the teal column on the left side of the table. The second letter can be found in the teal row all the way on the top of the table, and the third letter is in the teal column at the right side of the table. Let's say you have the codon AUG. Remember, we go to the left column for the first letter. The left column has the letters U, C, A, and G. Notice that all the codons next to the letter U start with U. All the codons next to the letter C start with C. Are you beginning to see a pattern here? So we need the third group of codons, those all starting with the A nucleotide. Okay, on to the second nucleotide in the AUG codon. It is the letter U, and the second letter is found looking at the teal row all the way on top of the table. It has the letters in this order: U, C, A, G. Each letter is at the top of a gold column. We want the letter U, so we look at the first gold column. All the codons in this column have the letter U as their second letter. Now we look where this column intersects the row we identified in the previous step. We find one cell where all the codons start with the 2 letters AU. There are only 4 codons in this cell: AUU, AUC, AUA, and AUG. Our codon is AUG, and the amino acid next to it is abbreviated MET. MET stands for the amino acid methionine. So the amino acid corresponding to the AUG codon is methionine. Let's do another one, the codon AGU. We go to the third row for the A and then to the fourth column for the G. At the intersection of these two, we find a cell in which all 4 codons start with AG. The third letter is shown in the teal column on the right side of this cell, in the order U, C, A, G, from top to bottom. AGU is the first codon in this cell, and it stands for the amino acid SER or serine. You can see that the next codon down, AGC, also codes for serine. There are 64 codons in the genetic code and only 20 amino acids. So most amino acids are coded for by more than one codon. There are actually 3 codons that do not code for any amino acid. They are the stop codons. As the name implies, they are used to indicate the end of the polypeptide. The 3 stop codons are UAA, UAG, and UGA. The statement that the genetic code is universal means that the same genetic code that we saw in the last slide on that table is used by all organisms. There are a few minor exceptions, say in mitochondrial DNA and in some parasitic protozoans, but basically all organisms use that same code. That also means that you can take a gene from one organism and express that gene, make the protein that the gene codes for in another organism. If the code were not the same in both organisms, then, a gene from one organism would be pure gibberish in the other, but it can actually be interpreted in the other organisms. And that is what makes it possible for humans to make recombinant DNA that has genes from other organisms inserted into DNA from a given organism. And for example, you can have E. coli bacteria produce human insulin, and you can use a tobacco plant to produce vaccines for humans or antibodies also for humans. You can make crops such as golden rice that have a gene that codes for the metabolic pathway to produce beta carotene in rice, so that you can make rice that when people eat it, they can produce vitamin A. Or you can make canola, another crop, to produce laurate, which is a fatty acid that is usually not found in that crop. So the universality of the genetic code makes all of this possible. Here are some more examples of how the genetic code is universal and how you can express genes from one organism in another one. On the left side, we see a little tobacco plant that has been engineered to contain a firefly gene that is responsible for the production of light. Again, this is like allows that reaction to proceed, and the plant itself can light up. And here on the right side is a pig that has been engineered to contain a gene from a jellyfish originally that fluoresces. In this case, the fluorescence is yellow, so you can see in the pig's nose and in the hooves, you can see the yellow color. Naturally, the fluorescence used to be green, but they have modified that as well, as you will see on the next slide. Here we see some fish that are known as glowfish, And glowfish are fish that are derived from this fish, the zebrafish. And this is the natural color of the zebrafish. It's got blue and white stripes. The glowfish have been genetically engineered to have that gene from the jellyfish in them, and that the green fluorescent protein. But the green fluorescent protein gene itself has also been modified so that it reflects the energy at different wavelengths. So now you've got yellow fluorescent protein, red fluorescent protein, orange, and green. So you have a rainbow of colors possible. This isn't done simply for entertainment, although these fish are sold as pets in some states. But the green fluorescent protein is used as a marker, and it's used a lot in laboratories to indicate where certain proteins are located in cells, or if a cell is migrating during the process of development. For lots and lots of different investigations in biology, it is a valuable tool. Now we're going to take a closer look at the process of transcription. Remember that it entails the synthesis of an RNA molecule using DNA as the template. It is done or performed by a protein called RNA polymerase that is represented by this over here. RNA polymerase binds to a location on the DNA that's called the promoter. The promoter is basically the place on the DNA that tells RNA polymerase, start synthesizing right after me. So RNA polymerase binds to the promoter and then starts transcribing right after that. The RNA is synthesized in the 5' to 3' direction. This is the same direction as DNA replication occurs. In generally, synthesis of nucleotides always occurs in the 5' to 3' direction. The transcription process is divided into 3 main parts: initiation, elongation, and termination. Initiation involves the binding of RNA polymerase, of that protein, to the promoter. And remember, the promoter is a location on the DNA right in front of the gene. It also includes the addition of the first RNA nucleotide. So this is when the transcription is going to begin. RNA polymerase does not need a primer. This is different from DNA polymerases, which need both a template and a primer. After initiation comes elongation. The same RNA polymerase is going to keep adding new nucleotides going in the 5' to 3' direction. In other words, it always adds to the 3' end. And in doing that, RNA polymerase is going to unwind the DNA that is right in front of it and rewind it behind it. And the place where the 2 DNA strands are actually separated is called the transcription bubble. The transcription bubble is relatively small, maybe 17 to 18 base pairs are separated in that bubble. Then comes the termination part, in which RNA polymerase is going to release the transcript, releases the RNA, and then it detaches from the template, it detaches from the DNA. We're looking at a more detailed picture of the transcription process, showing the transcription bubble. The DNA template are the blue molecules. So you've got this and this is the double-stranded DNA that has been separated. So the bubble goes from here to there, and you have these separated bases. The template is the lower DNA strand, so this part here is what is going to be copied by RNA polymerase to produce the RNA transcript. The transcript is shown in red. Notice that here is the 3' end of the transcript. New nucleotides are added to that 3' end, so this A is just about to be added, and it's going to use that T from the template to choose that the A is indeed the correct one to be added, and then it's going to be attached to the previous nucleotide, forming a phosphodiester bond between the two. And the next one coming up is going to be a U, which is going to pair with the A over here. And RNA polymerase protein takes care of all of that, and at the same time it unwinds the DNA in front of it and it rewinds the DNA behind it. In eukaryotes, the initiation of transcription is similar to what we saw already in prokaryotes, a little bit more complicated. And in general, that's the truth when you compare eukaryotes with prokaryotes. In general, the RNA polymerase protein, the RNA polymerase that performs the transcription, in eukaryotes, it doesn't directly recognize the promoter. Instead, the promoter is going to be recognized by these accessory proteins known as transcription factors. So the transcription factors are going to interact with the promoter, in this case with an area on the promoter that's called the Tata box, and the name is the Tata box because of that sequence that we have here. So the transcription factors recognize the Tata box and bind to the promoter, and then RNA polymerase 2 recognizes the transcription factors and sits down, and then the process of transcription can begin. A little comparison between prokaryotes and eukaryotes. In prokaryotes, in bacteria, we only find one RNA polymerase. In eukaryotes, there are at least 3 different RNA polymerases. If you take genetics, you'll learn there's 5 different RNA polymerases. In prokaryotes, you see that the RNA polymerase itself will interact with the promoter on the DNA. In eukaryotes, instead of RNA polymerase interacting with the promoter, it's transcription factors that bind to the promoter first. They bind at the Tata box, and then they call RNA polymerase to the promoter. They attract RNA polymerase to the promoter, and then they form something called a transcription initiation complex. In prokaryotes, there are actually specific sequences that indicate where transcription is supposed to finish, and they're called terminators. In eukaryotes, we don't really have those terminators, so the actual location of the termination is going to vary as the RNA polymerase protein continues to transcribe after the end of the gene, and there's going to be another protein that is basically going to degrade the transcript as RNA polymerase is still transcribing, and that is how it's going to end. The making of the RNA transcript is only an intermediate step. We want to make a protein, and that is going to be accomplished in the process of translation. So translation is the making of a polypeptide chain, this time using messenger RNA as template. The organelle that is going to do this, it has the status of a whole organelle, is a complex that is called the ribosome. And I just circled the ribosome is the brown structure. It's actually composed of two parts. So this is the small subunit, and this is the large subunit of the ribosome. The information that is being translated is found on the messenger RNA molecule. We see that here in red. So the messenger RNA is going to be read by the ribosome. The message is in the form of nucleotide triplets that are called codons, and each codon specifies one amino acid. Now, how are these amino acids going to get to the ribosome? They're going to be brought there by a different RNA molecule called transfer RNA. The transfer RNAs are shown here in this teal blue. Each transfer RNA is going to bring an amino acid. We see one here. The transfer RNA is the green-blue structure, and then here's the amino acid. So a transfer RNA brings an amino acid to the ribosome. A very important part of the transfer RNA is this anticodon. The anticodons are shown over here, this part. An anticodon is a triplet of RNA nucleotides. It is antiparallel and complementary to the codon found on the messenger RNA. Remember, the messenger RNA consists of these codons. You've got 3 nucleotides form one codon, and then each individual transfer RNA is going to be complementary to one codon. So one anticodon binds to one codon on one side of the transfer RNA, and on the other side of the transfer RNA we see that it carries an amino acid that is going to be incorporated into a polypeptide chain, and we see that over here, and that is all accomplished by the ribosome. The next few slides are dedicated to transfer RNA. Remember, transfer RNA is the molecule that is going to form the connection between the message on the messenger RNA codons and the amino acids that are going to form part of the protein. Each transfer RNA brings one amino acid to the ribosome. There are about 45 different kinds of transfer RNA in a cell, and all of them are about 75 to 80 nucleotides long, so they're fairly similar to each other. They all fold into a 2-dimensional shape and then a 3-dimensional shape. That this folding is made possible by the transfer RNA binding to itself through hydrogen bonding at places where it is complementary to itself. And we're going to see that on the next slide. Every transfer RNA has an anticodon, in other words, 3 nucleotides that are going to pair with the codon on the messenger RNA. And on its other side, it has an amino acid attachment site. And that amino acid attachment site is always CCA, going from 5' to 3'. There are small differences in sequence and in structure that are needed to make each tRNA unique. These are the different representations of the transfer RNA. The cloverleaf structure is the 2-dimensional structure, and then the L shape is the 3-dimensional structure. The L is upside down, and then this little sausage is is how the book draws them. So let's take a look at the cloverleaf shape, because that shows us the actual basis. We see the anticodon over here. In this case it's AAG, but the anticodon is going to be different depending on what codon it's going to bind to. So that's going to be varying. Then you've got the amino acid attachment site, the CCA, that you find on all of them. And notice that parts of the molecule are complementary to itself, so that you have hydrogen bonding between these bases over here. You see this? Hydrogen bonds and hydrogen bonds here. And you've got more hydrogen bonds there. So those hydrogen bonds will cause the formation of that cloverleaf shape, and then you have different arms in the molecule. Now the cloverleaf is going to fold upon itself and give you the 3D structure, which is the L shape. Notice the color coding. You've got a pink loop here, and you have a blue loop over here, and a green loop. So the pink loop and the blue loop are going to be interacting, they're sort of superimposed on each other. If you just imagine the molecule folding so that the pink loop and the blue loop are on top of each other, and now this part is bending to the right, so you get that the amino acid attachment site ends up over here, while the anticodon ends up there. That makes it possible for the amino acid attachment site to be as far away as possible, as far as the actual distance between the two. And then here's our book's representation. It shows us the anticodon. Remember, that is the part that is going to interact with the messenger RNA, and the anticodon is both antiparallel and complementary to the codon. And then here is the amino acid attachment site, that is this part over here. We saw that the anticodon is an intrinsic part of the structure of each tRNA. But how does the tRNA get its amino acid? It has to be loaded with the amino acid, or charged with the amino acid, by a special protein that is called aminoacyl tRNA synthetase. And there are 20 different ones, 20 different aminoacyl tRNA synthetases, one different one for each amino acid. And each of them will recognize all the tRNAs that carry the same amino acid. Remember, you saw in an earlier slide that there are about 45 different tRNAs. There are only 20 different amino acids, so that also means that some tRNAs will carry the same amino acid. These tRNAs are known as isoaccepting tRNAs, and all tRNAs that accept the same amino acid are going to be charged, loaded, by the same aminoacyl tRNA synthetase. The process of charging a tRNA with its correct amino acid is shown in this slide. We see the enzyme that performs this process, an aminoacyl tRNA synthetase. In this specific case, we're looking at tyrosyl tRNA synthetase. It is called that because the amino acid that it will charge to the correct tRNA is the amino acid tyrosine, and that's shown over here. The tyrosine tRNA that is going to be charged is here. Notice how the shape of the enzyme has these binding sites where both of the substrates fit, the RNA, the tRNA, and the amino acid. In the second part of the illustration, we see how the aminoacyl tRNA synthetase is going to hydrolyze ATP to provide the energy to bind both of its substrates, attach the amino acid to the tRNA, And then in the third step, we see the charged tRNA that is released, the amino acyl tRNA, in this case now, charged with the amino acid tyrosine. Over here, we see a more realistic model of the shape of both the amino acyl tRNA synthetase, shown in blue, and then you see the tRNA. Can you recognize that L shape that we looked at earlier? The tRNA, how it's binding to the enzyme, and here is the amino acid as well binding to the enzyme. Let's review a couple of these important facts about tRNA and mRNA. tRNA, transfer RNA, has an anticodon as part of its structure. And the anticodon is a triplet, a 3-nucleotide sequence. And this triplet is complementary to another triplet that is found on the messenger RNA. And the triplet that we find on the messenger RNA is called the codon, and is actually what is in that table of the genetic code. There's all the codons on different messenger RNAs. Now here we learn something interesting. The codon to anticodon pairing is flexible. So one transfer RNA can sometimes pair with more than one messenger RNA, and that characteristic has been called wobble. And the wobble occurs at the 3' end of the messenger RNA. In other words, the 3' end of the messenger RNA can sometimes vary so that you can have messenger RNAs that have a different nucleotide at the 3' end, and they can bind to the same tRNA anticodon. Here is an example of that. You've got the tRNA, and it has the anticodon from 3' to 5' UCU. It is able to pair with AGA on an mRNA codon or with AGG on an mRNA codon. So while usually it is U with A that should pair, here you see a pairing of U with G on the last nucleotide in the mRNA codon. That's the wobble part of the binding. And it explains why you can have 61 codons that code for an amino acid that are binding to only 45 different tRNAs. So that means that some of those tRNAs are binding more than one mRNA codon. Now it's time to take a look at the organelle that is responsible for the process of translation or protein synthesis. It is the ribosome. So the ribosome is that cellular organelle that makes the proteins. In fact, we learned that back in chapter 6 when we were first looking at cellular organelles, and now we're going to take a closer look. A ribosome's composition includes RNA, specifically ribosomal RNA or rRNA, and proteins. The ribosome has to interact both with messenger RNA and with tRNA. And in order to do that, for the binding for the tRNAs, it has 3 different binding sites for tRNAs. One is called the exit site, or the E, and that is for tRNAs that have already given up their amino acid. They're discharged, and they're on their way out of the ribosome. Next to the E site comes the P site. The P stands for peptidyl, and it includes, or it is for the tRNA that is carrying the whole polypeptide, the growing polypeptide. And then we have the A site, or aminoacyl site, which is for the incoming tRNA. So the way that these were presented is sort of in reverse order. You're going to have the aminoacyl site, and then it's going to go to the P site, and then it's going to go to the E site. The ribosome catalyzes peptide bonds for every protein. The little asterisk here tells us that there are a few exceptions, but we're not going to worry about that in Bio 210. Remember that a peptide bond is a covalent bond between two amino acids, between the carboxyl end of one amino acid and the amino end of another amino acid. A sort of realistic view of the shape of a ribosome. It has a large subunit, which is shown in blue, and a small subunit in beige. And then the messenger RNA is shown as these beads, these yellow beads over here. The messenger RNA is basically interacting at the interface of the two subunits. The three binding sites for transfer RNAs are here, and we actually have 3 transfer RNAs occupying those binding sites. Remember, the E site, the P site, and the A site. Notice that there is this blue bead, set of beads coming out, and that's the polypeptide. Each little bead represents an amino acid coming out of the exit tunnel of the ribosome. Keep in mind that for the ribosome, whether the small subunit or the large subunit is on top doesn't really matter. Probably they're rotating in three-dimensional space. So in some illustrations you will find it with the small subunit on top, and sometimes you'll find it with the large subunit on top. Now this is a more cartoonish view, but sometimes they show the relationships between components better. So we're looking again at a ribosome, and we see the small subunit here and the large subunit over here. We see the messenger RNA in red, and we see how it is sort of located between the two subunits of the ribosome. The 3 binding sites for tRNAs are here. One of them is now not occupied, so you see it's got the E. The P site is this one, and the A site is that one. So both the P and the A site are occupied at the moment. Notice that the tRNA that is in the P site is carrying the growing polypeptide. So it's carrying the whole polypeptide that has been synthesized so far. And then the one on the A site is carrying the latest amino acid, the one that is now its turn to be added to the polypeptide chain. And we're going to see in the upcoming slides how this process is accomplished. Like DNA replication and transcription, translation is divided into 3 stages called initiation, elongation, and termination. During initiation, the messenger RNA and the first tRNA, which is already charged with the amino acid methionine, and the ribosome are needed. There are also initiation factors that are proteins that are going to help with getting these three components together. The start codon on the messenger RNA, which we remember is AUG, is going to establish the reading frame of the polypeptide. This initiation process is going to use 1-GTP. Now, GTP is a molecule very closely related to ATP. Instead of adenosine triphosphate, it's guanosine triphosphate, and its hydrolysis is a little bit more energetic than that of ATP. And in general, for the process of translation, we use GTP instead of ATP, translation at the ribosome. So that was the placement of the very first amino acid of the protein. Then comes elongation, and in elongation, the messenger RNA is going to be moved, or the ribosome moves with respect to the messenger RNA, and a peptide bond is going to be formed between the latest amino acid that has been inserted and the previous one. When the ribosome moves, it's called translocation. So the ribosome moves on the messenger RNA. When this happens, there's going to be a new A site that is going to be free. So a new transfer RNA is going to enter that A site. The old discharged tRNA is going to enter the E site from the P site where it was before, and from the E site it leaves the ribosome. Each elongation cycle, at each elongation cycle we're going to add one amino acid, and it's going to use 1-GTP. And the elongation cycle is going to be repeated as many times as there are amino acids remaining in that polypeptide. Then comes termination. Termination is going to occur when the ribosome reaches one of the three termination codons. We saw in the genetic code that they are UGA, UAA, and UAG. There are proteins called release factors that are going to assist with the process of termination, and termination is going to use up 2 GTPs. So let's take a look at the initiation of translation. In the first part, we see coming together the small subunit of the ribosome with the messenger RNA and the first tRNA, the one that is charged with methionine. The little inset here shows us how the anticodon on the tRNA is complementary to the codon on the messenger RNA, and there are hydrogen bonds between them. The messenger RNA codon from 5' to 3' is AUG. The tRNA anticodon from 5' to 3' is CAU. Remember, if we're going from 5' to 3' on the anticodon, we'll be going from right to left. This complex is going to need the hydrolysis of a GTP to recruit the large subunit of the ribosome, and now we're going to be ready for adding more amino acids. So now you've got the translation initiation complex formed, and we see how the first amino acid is going to be located. The tRNA that carries the first amino acid is located at the P site of the ribosome. When we get to the process of elongation, we'll see that every subsequent tRNA coming into the ribosome is going to enter in the A position. Elongation is a series of steps that goes in a cycle. It includes the recognition of the codon, the formation of a peptide bond, and the movement or translocation of the ribosome. On the top, we see the first step, the recognition of the codon, and that recognition is between the codon of the messenger RNA and the anticodon of the transfer RNA. So the new transfer RNA is going to come into the A position of the ribosome. It'll take hydrolysis of GTP to place it. So now the new transfer RNA is sitting in its position in the A site of the ribosome, and then the previously synthesized polypeptide is being carried on the transfer RNA that's on the P position. At this point, we're going to see that the tRNA in the P position is going to transfer the whole polypeptide to the tRNA on the A position. If we go from here to the next drawing, you see in this one it is the tRNA in the P position that's carrying the big polypeptide. Notice now it's the one in the A position, and that those 4 amino acids that were there already are added to the last amino acid, the latest amino acid that is carried on the amino acid, on the tRNA on the A site. Okay, now we're going to hydrolyze another GTP molecule, and that is going to allow the ribosome to move. So the ribosome is going to move so that now the A site is open. You see that? The ribosome moved in this direction, and the A site is now open, and the tRNA that used to be in the A site is now automatically in the P site, and the one that was in the P site is now in the E site, and from the E site it gets booted out of the ribosome. So that one goes out. You can look at this as either the ribosome moving to the right, or you can look at it as the mRNA moving to the left. Both of those accomplish the same thing. At this point now, the ribosome is going to be ready for the next amino acid, the next tRNA carrying an amino acid, so then the cycle begins again. Termination happens only once for each polypeptide. At one point, the A site of the ribosome is going to encounter a stop codon on the mRNA. That could be either UAG, UAA, or UGA. There is no tRNA that fits to that stop codon, that has complementarity to that stop codon. Instead, there is a protein called a release factor that recognizes the stop codon, and the release factor is going to insert itself in the A site of the ribosome, almost as if it were a tRNA. But the release factor does not carry any amino acid. At this point over here, you're going to see that the tRNA that is in the P site is going to transfer its polypeptide to a water molecule on this release factor, and basically that is going to release the polypeptide from the ribosome altogether, And you're going to use some GTP to facilitate these reactions. And at this point, the party is over and everybody is going to disperse. The subunits of the ribosome separate from each other. The messenger RNA is going to be released. And this messenger RNA can be reused to code for another copy of the same protein. And also the ribosome subunits can be reused to translate any other protein that needs to be translated. In chapter 5, we learned that each polypeptide has an N terminal or an amino terminal, and it has a C terminal or a carboxy terminal. And the synthesis of a polypeptide goes from the N terminal to the C terminal. So the N terminal is made first, and the C terminal is the last one. The last amino acid is the one at the C terminal. The same messenger RNA can be simultaneously translated by several ribosomes at the same time. Then you form a structure called a polyribosome. And we see that here the messenger RNA is shown in red, and you see 4 ribosomes that are busy translating that messenger RNA, and they're starting on the left side and going to the right side. Over here is one ribosome that has finished, so the subunits have separated from each other. From left to right, we can see the protein getting longer as it exits the ribosome, and then here, the last one, the protein has been completed. On the bottom, we see an actual electron micrograph that shows that same process. So you see here a messenger RNA with multiple ribosomes attached to it. The correct folding of a protein helps it to achieve its active conformation, so it needs to assume the correct tertiary structure. Depending on the kind of protein that it is, that folding is going to happen either in the endoplasmic reticulum or in the cytoplasm. In the cytoplasm, it can be helped by the special proteins called chaperonins. So how does a protein get to its correct location? If it's a protein that's going to be a membrane protein, or if it's part of the endomembrane system, it is going to have a signal peptide. So a certain series of amino acids that are part of the polypeptide, and that signal peptide will be recognized by a signal recognition particle that is going to guide the whole assembly to a signal recognition particle receptor protein on the endoplasmic reticulum. And we'll see how that comes together in the next slide. The protein is then going to be threaded through a pore in the endoplasmic reticulum that is known as a translocation complex. We're using that word translocation again. We used it just before, earlier, to talk about the movement of the ribosome during elongation of protein synthesis, and here we're using it to refer to the threading of the protein through the endoplasmic reticulum. Translocation really just means movement. So in the ER, once a protein is inside the endoplasmic reticulum, the signal peptide is going to be taken off. So this is where you're going to get that methionine amino acid is going to be removed, the proteins that are going to be part of the membrane are first going to be embedded in the endoplasmic reticulum membrane, and then they're going to move to their final target membrane in the endomembrane system. If it's a protein that's supposed to be secreted, then that protein goes to the lumen of the endoplasmic reticulum. In other words, the interior of the endoplasmic reticulum. It's not going to be embedded in a membrane, so that when a vesicle that butts from the endoplasmic reticulum makes it to the plasma membrane and fuses with the plasma membrane, whatever is inside that vesicle will then be secreted from the cell. Let's take a look at that process of bringing the ribosome to the endoplasmic reticulum. Here is our ribosome that is actually still in the process of making the protein, and the protein has a signal peptide. The first 20 amino acids, or about that number, that have been synthesized on the protein form that signal peptide. It is shown here in green. The signal peptide is recognized while still in the cytoplasm by a signal recognition particle, SRP. And that signal recognition particle, in turn, is going to be recognized by a signal recognition particle receptor protein, which is here. And when those two, the signal recognition particle and the signal recognition particle receptor protein, when they attach to each other, the whole ribosome is now transferred to the rough endoplasmic reticulum. Remember, the rough endoplasmic reticulum is the ER that is associated with ribosomes, and that is why it was called the rough endoplasmic reticulum. So now the ribosome has arrived at the rough endoplasmic reticulum, and here it is very firmly attached. The signal recognition particle is going to move off to recognize another signal somewhere else, and here we see the protein is being threaded through that translocation pore into the endoplasmic reticulum. The signal is still attached to it, the signal peptide, and then as the protein gets bigger, the signal peptide is removed, and then finally, when the protein is finished, it is released into the lumen of the endoplasmic reticulum, and the ribosome is going to dissociate, and the mRNA is released, and it can be used again. So this protein is likely going to be a secreted protein because it has not been embedded in the membrane of the ER. If it was going to be a membrane protein, it's going to be embedded in the ER membrane, and then from there it's going to end up in a vesicle. Here it's going to end up in the lumen of the vesicle, and then it's going to be secreted from the cell. When a protein is made, when translation occurs, you make the primary structure of the protein. You're going to put one amino acid together with another amino acid. After that has been finished, for the protein to be active, besides it folding into its active conformation, there are some post-translational modifications that have to be made to most every protein, and they're needed for that protein to be active. They're going to be different for each different protein, and they include the addition of sugars or glycosylation, the addition of lipids, the addition of phosphate groups to the protein in specific positions. They could sometimes include the cleavage of a polypeptide as is the case with insulin, which is made as one large protein which gets cleaved into two separate polypeptides, and then those polypeptides are going to get connected by disulfide bridges. Remember the sulfhydryl functional group that we studied in chapter 5? Two of those together form a disulfide bridge, and they are used in proteins. Another thing that can happen to proteins is that they may have quaternary structure, in which you have more than one polypeptide chain for a protein. In other words, two or more polypeptide chains come together from genes that were translated separately. And hemoglobin is an example of that. We've got alpha hemoglobin and beta hemoglobin, and our regular hemoglobin protein has two alpha subunits and two beta subunits that are translated from different mRNAs that come from different genes, and then they're going to be put together as a functional hemoglobin protein during post-translational modification.